Welcome to video series on protein metabolism. This video is assets. What you will learn in this video. Histidine, alanine, serine, glutamate and branched-chain amino acid pathway in a simplified way. Biochemical basis of anaphylaxis, maple syrup urine disease, Cahill cycle using flow charts and its associated clinical correlation. Important MCQ discussion, probably university exam and memory. Why is metabolism of histidine, alanine, serine, glutamate? That will be asked. Describe metabolism of histidine, explain glucose-alanine cycle, discuss biochemical basis of allergic reactions or anaphylaxis. What is the significance of FIGLU? Describe metabolism of branched-chain amino acid. From patient point of view, you need to know the biochemical basis behind allergic reactions, anaphylaxis, maple syrup urine disease, and urocanic acid. Now coming to metabolism of histidine. Histidine is acted upon by histidase enzyme wherein the amino group is released and this results in the formation of urocanic acid. Now this undergoes hydration catalyzed by the enzyme urocanase to form imidazole propionic acid. This is acted upon by hydrolase enzyme forming form-amino-glutamic acid. Now the form-amino group is donated to tetrahydrofolate and it gets converted to glutamate. So now this glutamate gets converted to alpha ketoglutarate by entering into TCA cycle, gets converted to succinyl CoA, which gets converted to succinate, fumarate, malate, oxaloacetate, and this oxaloacetate gets converted to pyruvate, and pyruvate by gluconeogenesis gets converted to glucose. That's the reason why histidine is glucogenic in nature. Okay, so this can be asked as a 5-marker question or it can be asked as a 3-marker question. Now the other important question that will be asked is what is FIGLU? FIGLU is form-amino-glutamic acid. And what is the importance of FIGLU? So normally what happens, this folic acid accepts the form-amino group from form-amino-glutamic acid and releasing glutamate. So whenever there is deficiency of folic acid, what happens? This glutamate is not formed and form-amino-glutamic acid will accumulate in the patient body and it gets excreted in urine. So FIGLU, that is form-amino-glutamic acid excretion, is an indicator of folic acid deficiency. Now, histidine undergoes decarboxylation to form histamine. So, histamine is a biogenic amine. Now, this histamine is formed in platelets, mast cells, and basophils. Now, histamine has got a variety of biochemical or biological effects on variety of organ systems. On cardiovascular system, it causes hypotension and tachycardia. On respiratory system, it causes bronchospasm. On blood vessel, it causes dilatation, and this has been implicated whenever the superficial temporal artery vessels are dilated. This has been implicated in the precipitation of migraine attacks. And histamine also stimulates gastric acid secretion. So histamine is also responsible for allergy and anaphylactic reaction, and the biochemical basis is as follows. This can be asked as a 3-marker question or a case-based question also. So whenever allergen enters our body, now this allergen is handled or tackled by immunoglobulin E. Now this allergen along with this immunoglobulin E goes and fixes on the mast cells. Okay, so subsequently it causes degranulation of the mast cells, and this mast cells contain histamine and also slow releasing substances, so reacting substances of anaphylaxis. So all these will result in hypotension, tachycardia, bronchospasm and blood vessel dilatation, and this are known as this is how the allergy or the anaphylaxis gets managed. Just to summarize, nature contributes to one carbon pool by donating formamino group to folic acid, and the imidazole group of histidine has got a pK of 6.1, which is close to physiological blood pH, and because of this it has got a maximum buffering capacity. So two important proteins have got large amount of histidine residues. For example, albumin has got 16 histidine residues and hemoglobin has got 38 histidine residues, and that's the reason these two proteins have got maximum buffering capacity when compared to other proteins. Histidine undergoes decarboxylation to form histamine, and this is responsible for allergy and anaphylactic reactions. So there are two important disorders associated with histidine metabolism. One is histidinemia and the other one is urocanic aciduria. When histidase enzyme is deficient, it results in histidinemia. So normally histidine gets converted to urocanic acid by histidase enzyme, and this enzyme is deficient. It undergoes transamination to form imidazole pyruvic acid, and this gets excreted in urine. So apart from that, this autosomal recessive disorder characterized by mental retardation and delayed speech. The other enzyme that is deficient is urocanase enzyme. Normally urocanic acid gets converted to imidazole propionic acid with the help of urocanase. When this enzyme is deficient, there occurs accumulation of urocanic acid and it gets excreted in urine, resulting in urocanic aciduria. So this is the summary of histidine. It is semi-essential amino acid. It is glucogenic, contributes to one carbon pool. It acts as blood buffer because of the presence of imidazole group which has a pK of 6.1, and this is close to physiological blood pH. It is responsive. Histidine undergoes decarboxylation to form histamine, which is responsible for allergy and anaphylaxis. Disorders include histidinemia, histidase deficiency, urocanic aciduria, deficiency of urocanase. Okay, so this can be asked as a 3-marker reasoning based question. Why does histidine act as a blood buffer? This is because the pK of imidazole group of histidine is 6.1 and this is close to physiological pH. Coming to metabolism of branch amino acids, there are 3 branch amino acids: valine, leucine, isoleucine. So valine is 5 carbon compound, leucine is 6 carbon compound, and isoleucine is also 6 carbon compound. All the 3 branch amino acid undergo transamination to form respective keto acids. Valine gets converted to alpha ketovaleric acid. Now leucine gets converted to alpha ketocaproic acid. Isoleucine gets converted to alpha keto methyl valeric acid. There is no other go, you have to just memorize these substrates. In the next step, all the 3 keto acids of all the 3 branch amino acid undergo oxidative decarboxylation. So wherein valine gets converted to isobutyryl CoA, leucine gets converted to isovaleryl CoA, isoleucine gets converted to methyl butyryl CoA. So how to remember this in a simplified way is, valine is a 5 carbon compound. After undergoing oxidative decarboxylation, it gets converted to 4 carbon compound, and as you all know, 4 carbon compounds are butyryl compounds. Okay, that is a easy way to remember. Next, leucine is 6 carbon compound. After undergoing oxidative decarboxylation, it gets converted to 5 carbon compound, and 5 carbon compound is valeryl compounds. Okay, isoleucine is also 6 carbon compound. It undergoes oxidative decarboxylation to form 5 carbon compound. Here its 4 carbon compound is butyryl. To when methyl group gets attached, it becomes methyl butyryl CoA. Okay, 5 carbon compound. So subsequently, by a series of reactions, this valine finally ends up with formation of succinyl CoA. So very difficult to remember all these intermediate steps. So in exam, you can skip these steps and you can directly write succinyl CoA. And as you all know, succinyl CoA enters TCA cycle, gets converted to succinate, fumarate, malate, oxaloacetate, pyruvate, and pyruvate gets converted to glucose by gluconeogenesis. So valine is only glucogenic in nature. Coming to leucine characteristic feature, it gets converted to acetyl CoA and acetyl CoA are the end products of leucine metabolism. And as you all know, both of these are ketone bodies. So leucine is purely ketogenic amino acid. Coming to the end product of isoleucine metabolism, it ends with the formation of acetyl CoA and propionyl CoA. As you all know, acetyl CoA is ketogenic and it will enter ketogenic pathway. Propionyl CoA gets converted to succinyl CoA. It will enter into TCA cycle and gets converted to pyruvate, and pyruvate gets converted to glucose. So it is glucogenic. So only isoleucine is both glucogenic and ketogenic. So I repeat, valine is purely glucogenic. Leucine is purely ketogenic, whereas isoleucine is both glucogenic and ketogenic. Okay, so one important inborn error of metabolism associated with branch and amino acid is maple syrup urine disease, and this can be asked as a 5-marker or a case-based question. Discuss maple syrup urine disease. So the conversion of the oxidative decarboxylation of keto acids is deficient here. So keto acids will be accumulated in the child's body and they get excreted in urine. So, and this urine has got burnt sugar odor and hence the name maple syrup urine disease. So mnemonic to remember this is DREAM, DRM cube. Okay, so E and A silent here, DRM cube, DREAM cube you can remember. D stands for deficiency of branch chain keto acid decarboxylase. R stands for Rotheras test is positive because they are keto acids, and M cube stands for maple syrup odor of the urine, burnt sugar odor, or easy way to remember is maple syrup odor of urine. And the child will be mentally retarded, so mental retardation is also a characteristic feature. And metabolic acidosis occur because all the keto acids are getting accumulated in the child's blood. So this is how you can remember maple syrup urine disease. Coming to quick summary of this branch and amino acid, all of the three amino acids, valine, leucine, isoleucine, are essential amino acid and they have to be consumed in the diet. They have an important role in muscle energy metabolism. So muscles preferably utilize branch and amino acids for energy purpose. Valine is glucogenic, leucine is purely ketogenic, and isoleucine is both glucogenic and ketogenic. So important disorder you need to remember is maple syrup urine disease due to deficiency of branch and keto acid decarboxylase. There occurs accumulation of this keto acids resulting in metabolic acidosis, mental retardation, and burnt sugar odor, and Rotheras test will be positive. Coming to alanine, alanine is a non-essential amino acid. It is synthesized from pyruvate by transamination. So pyruvate is a keto acid. It accepts the amino group from glutamate, which gets converted to its keto acid alpha ketoglutarate, and pyruvate, after receiving the amino group, gets converted to alanine. So this most important amino acid taken from the peripheral tissue. So this is the most important transamination reaction and catalyzed by the enzyme alanine transaminase. It requires pyridoxal phosphate active form of B6 as coenzyme. Now this has got a great clinical significance also because alanine transaminase is one of the enzyme used to assess the liver function capacity also. So alanine is also synthesized from glycine. So glycine transfers the methylene group to tetrahydrofolic acid and gets converted to glycine accepts the methylene group from folic acid and gets converted to serine. It undergoes dehydration to form pyruvate. Pyruvate undergoes transamination to form alanine. So alanine is also formed during tryptophan metabolism. Tryptophan gets converted to N-formyl kynurenine by pyrrolase enzyme. Then formyl group is donated to folic acid and it becomes kynurenine. So this is acted upon by kynurenase enzyme to form 3-hydroxyanthranilic acid and alanine. This is how the alanine is non-essential amino acid synthesized from pyruvate, glycine and tryptophan. Alanine plays an important role in maintaining the blood glucose whenever we are starving, and this is known as Cahill cycle or glucose alanine cycle. This itself can be asked as a 5-marker question or a 3-marker question. So the explanation of Cahill cycle is as follows. Now, muscle will oxidize glucose for its energy purpose by glycolysis, and the end product of aerobic glycolysis is pyruvate. Pyruvate undergoes transamination to form alanine. So the mechanism is whenever we are starving, the muscle protein is broken down and all the amino acids get transaminated to form alanine. Now this alanine is released into general circulation, is taken up by the hepatocytes. This undergoes transamination to form pyruvate. The amino group is released as ammonia, which gets detoxified to urea, and pyruvate by gluconeogenesis gets converted to glucose. This is released into general circulation and reaches liver for muscle contraction purpose. So what you need to remember is from 2 molecules of alanine, 1 molecule of glucose and 1 molecule of urea is formed. So the main purpose of this glucose alanine cycle, also called as Cahill cycle, is to maintain the blood glucose in starvation. So the question can be asked as what is the significance of glucose alanine cycle? Just to summarize alanine metabolism, it's a non-essential amino acid synthesized from pyruvate, glycine, tryptophan, and alanine plays an important role in maintaining blood glucose, maintaining the blood glucose during starvation. The next amino acid is glutamate. It is also non-essential amino acid synthesized from alanine. Alanine donates the amino group to alpha ketoglutarate, which gets converted to glutamate, and alanine gets converted to its keto acid pyruvate. These are the functions of glutamate. Glutamate undergoes decarboxylation to form GABA, gamma aminobutyric acid, which is an inhibitor neurotransmitter in the brain. By accepting the ammonia, it gets converted to glutamine. So glutamate itself is an excitatory neurotransmitter in the central nervous system. Glutamate also contributes to the formation of glutathione by combining with glycine and cysteine to form tripeptide glutathione. And glutamate is glucogenic amino acid. This is because glutamate donates the amino group to oxaloacetate, which gets converted to aspartate, and it gets converted to its keto acid, that is alpha ketoglutarate, which will enter the TCA cycle, gets converted to succinyl CoA, fumarate, malate, oxaloacetate, pyruvate, and pyruvate gets converted to glucose by gluconeogenesis. That is how glutamate is glucogenic. So this is a quick summary of glutamate metabolism. It's non-essential amino acid, and for functions you can remember glutamate and G4. G4 for glutamate. So G4 stands for synthesis of GABA, glutamine, glutathione, and glutamate is glucogenic in. Coming to next amino acid, serine. Serine is a non-essential amino acid synthesized from glycine. So glycine accepts the methylene group from folic acid and gets converted to serine. And serine is glucogenic because serine undergoes dehydration to form pyruvate, and pyruvate by gluconeogenesis from glucose. See, that's the functions of serine. Serine is glucogenic, and serine forms pyruvate, and pyruvate can undergo transamination to form alanine. So serine can form alanine and serine can form glucose also. It undergoes decarboxylation to form ethanolamine. Ethanolamine can undergo methylation to form choline. So phosphatidyl ethanolamine and phosphatidyl choline are phospholipids that play an important role in maintaining, which are components of biomembranes. Apart from that, choline can combine with acetyl group from acetylcholine, and this acts as a neurotransmitter. And the other thing is phosphatidylserine. Apart from being a component of biomembrane, it is also a component of lung surfactant. Now serine can combine with cysteine and form selenocysteine, and this is the 21st amino acid. Now these are the functions of selenocysteine. It is present at the active center of glutathione peroxidase and deiodinase enzyme, and also thioredoxin reductase. Now, so these are the probable questions that can be asked. Which is the 21st amino acid? This is one of the most important Viva question that will be asked. So selenocysteine is the 21st amino acid. So this is also the Viva question, or it can be asked as a 3-mark question. What are the functions of selenocysteine, which is 21st amino acid? Now selenocysteine is present at the active center of glutathione peroxidase, which which will convert oxidized glutathione GSSG to reduced glutathione GSH. Then it is also a component of deiodinase enzyme, which is associated with conversion of tetrahydrothioronine to triiodothyronine. Then it is also present at the active center of thioredoxin reductase, wherein which is involved in the conversion of ribonucleotides to deoxynucleotides. So that was all about metabolism of alanine, serine, glutamate, branch chain amino acids, and histidine. Time for self-assessment. This question is from histidine metabolism. Which compound is formed directly from histidine and acts as vasodilator? Answer is histamine. Histidine undergoes decarboxylation to form histamine. Question on histidine degradation. Deficiency of which enzyme leads to increased Figlu excretion? And the answer is formiminotransferase. So this Figlu excretion is a marker of folic acid deficiency. Alanine is mainly involved in option urea cycle, glucose alanine cycle, ketone body synthesis, purine synthesis, and the answer is glucose alanine cycle, which transport nitrogen from muscle to liver, and this gets converted to urea and excreted by the kidney. The other important role of glucose alanine cycle is maintains the blood glucose whenever starving. Alanine is formed from oxaloacetate, pyruvate, alpha ketoglutarate, acetyl CoA. Answer is serine is a precursor of glycine. So dopamine is from tyrosine, histamine is from histidine, urea from carbon dioxide and ammonia, and serine from is glycine. Which vitamin is required for serine to glycine conversion? It is folic acid. So glutamate is a precursor of GABA. Serotonin is formed from tryptophan, histamine from histidine, melatonin again from tryptophan, and glutamate is a precursor of. So glutamate plays a key role in transamination reaction. Okay. So branch chain amino acids include all except lysine. Branch chain amino acids are mainly metabolized in muscle. Muscle has got a preference for branch chain amino acid for utilization for muscle contraction. So branch chain amino acid disorder, deficiency of branch chain keto acid dehydrogenase causes maple syrup urine disease. Okay. Phenyl ketonuria is due to deficiency of phenylalanine hydroxylase. Alkaptonuria due to deficiency of homogentic acid oxidase, and homocystinuria is due to deficiency of cystathionine beta. Glutamate combines with ammonia to form. Histidine is important for buffering capacity. Leucine is purely ketogenic. Okay, so this was all about the combined metabolism of histidine, alanine, serine, glutamate, and branch and amino acids. So just evaluate yourself and check out for the MC score. If your score is good, it is highly appreciable. Score is below 50%. So rewatch the video, and please resolve all the MCQs. Okay. So next video will be on polyamines and nitric oxide. Please do like, share, and subscribe for Dr. Preethi's Biochemistry Made Easy channel. Thank you.